This is the Krusty Lab, a podcast about molecular biology made by people with very different reasons to care about it. A molecular biologist, an experimentalist running cutting-edge research in what I can only describe as a shed, a venture capitalist who finds a funding opportunity in every cellular process, a professional skeptic who is usually correct, someone called Patar, and me. I'm Bobby Sponge. I'm the one who kept asking questions and got invited to stay. I want to tell you one thing before I introduce any of them. One thing. Right now, while you are listening to this, in every single cell of your body, and you have about 37 trillion of them, there are molecular machines reading a 3 billion letter message. You have zero awareness this is happening. The machines don't know you exist. They have been running every second of your entire life without a single conscious input from you. That message is your genome. And 8% of it, 8% of the code those machines are reading right now inside you is ancient viral DNA. Not a metaphor. Actual viruses that infected your ancestors millions of years ago got their genetic code permanently stitched into a reproductive cell and have been writing inside human chromosomes ever since. We inherited them. Most are silent, but some of them, the part I genuinely cannot get out of my head, some of them, evolution press-ganged into building the placenta. The structure that grows the baby, built partly from the remains of a virus. I found this out 6 weeks ago, and I have not fully recovered. This is episode 1. Let's start at the very beginning.
The framing is somewhat dramatic, but the facts are accurate. Welcome. Let's begin before Kelpie destabilizes something.
I already destabilized something this morning. It resolved itself, mostly.
Mostly resolved is not a phrase that should feature this early in an episode.
DNA, most undersold asset in the known universe. Let's fix the ignorance and see what monetizes.
My neighbor has a dog called DNA. I don't know if that's relevant.
It is not.
I suspected.
Dr. Seeley, absolute zero. What is DNA?
DNA is an information storage molecule. Every living organism on Earth uses the same system. Bacteria, trees, fungi, humans. Your body is running biological code. Your development, your physiology, the precise structure of every protein in every cell. All of it traces back to instructions encoded in DNA. 4 chemical building blocks. That's the entire vocabulary.
4.
Adenine, thymine, cytosine, guanine. A, T, C, G. The order encodes the biology. ATGCCA is a different instruction from ATCGCA. One wrong letter in the wrong position can silently do nothing, subtly alter a protein, or cause a genetic disease. The same molecule, the same 4 letters. Context is everything.
4 letters, all life on Earth. 4 letters.
Every software company I've funded needed at least 12 frameworks to do a third of that. I find this embarrassing.
Biology is absurdly over-engineered in some places, and laughably minimal in others. This is the minimal part. Hold on for the over-engineered part.
The 2 strands pair with strict complementarity. A always bonds T, C always bonds G. These pairs form rungs between the strands, which twist around each other into the double helix. The pairing rule is what makes copying possible. If you know one strand, you can reconstruct the other exactly. The structure is the replication mechanism.
A twisted ladder where each side tells you what the other side says.
Precisely.
Married couples are like that, except less accurate.
That is simultaneously off-topic and correct.
How long is this ladder?
The human genome is approximately 3.2 billion base pairs. Stretched end to end, the DNA in a single cell reaches roughly 2 meters. You have approximately 37 trillion cells.
I am not doing that maths.
I already did. 74 billion kilometers, past Pluto and back, more than once. I'm still working on the revenue model, but the raw number is viable.
2 meters in a thing I cannot see. How?
Extremely aggressive folding. DNA wraps around proteins called histones, molecular spools. That wrapping coils further into structures called nucleosomes, which condense further into chromosomes. The folding packs 2 meters of DNA into a nucleus roughly 6 micrometers across. But the folding is not just storage, it's control. Tightly folded DNA cannot be read. Loosely folded DNA can.
Which means the shape of the fold at any given moment in any given cell is a layer of information on top of the sequence itself. Same letters, different fold, different biology. I have been experimentally mapping this in a facility I operate independently.
In a shed.
I prefer low overhead independent research unit.
It is a shed, Kelpie.
The science inside is real. The structural classification is debatable.
Okay, 3 billion letters, 46 chromosomes folded into each cell. How much of it is actually doing anything?
Only about 1 to 2% of the human genome codes for proteins.
1%.
99% waste. In any company I fund, that would be a restructuring conversation.
Scientists called the rest junk DNA. That term is now considered incorrect. The non-coding regions contain regulatory sequences, structural elements, RNA genes, and the endogenous retroviruses Bobby mentioned at the start.
Wait, we're back to the viral DNA. Explain this immediately.
Over millions of years, retroviruses have infected germ cells, the cells that become eggs and sperm. Some integrated their DNA into the host genome and were inherited by all subsequent generations. These are endogenous retroviruses. In humans, they make up approximately 8% of the genome. Some are completely inert, others have been repurposed by evolution. One family is essential for forming the placenta.
The cell took ancient viral machinery and built a placenta with it. That is the single greatest example of hostile takeover I have ever encountered in biology.
We acquired a competitor's IP and deployed it internally. I've done this. It works.
It is mildly alarming that you are relating to a strategy pioneered by a retrovirus.
My grandmother said, never waste anything. She was very ahead of her time.
Okay, the junk is not junk. Got it. How does the cell actually read the code?
Transcription. The DNA itself almost never leaves the nucleus. It's too critical to risk. Instead, an enzyme called RNA polymerase binds upstream of a gene at a region called the promoter, unwinds the helix locally, and reads one strand as a template to build a complementary RNA copy, messenger RNA.
Photocopy the recipe. Never hand out the original.
Correct. And the copy is deliberately different from the original. RNA differs from DNA in 3 ways. It is single-stranded, it uses ribose sugar instead of deoxyribose, and it uses the base uracil, where DNA uses thymine. These are not arbitrary differences. The structural changes make RNA chemically suited to travel through the cytoplasm, where DNA would be degraded. The cell engineered a format specifically for export.
So DNA is the master copy in a vault. RNA is the working copy that leaves the building.
That is structurally sound, yes.
Our office does this with documents. Nobody ever explained it was also biology.
But not before processing. The raw RNA transcript has non-coding sections, they're introns, sitting between the coding sections. The spliceosome has to cut those out.
And the spliceosome is one of the most baffling machines in biology.
The spliceosome is over 150 proteins and 5 small nuclear RNAs. It completely reassembles itself on every single transcript from scratch and then disassembles again. Every single time. It's not a standing machine. It is a crowd of parts that forms a machine, does one job, and disperses. I tried to understand how it self-assembles for 3 months. I described the problem to my neighbor's cat. The cat understood nothing. I felt the same.
A bespoke factory, built for one item, demolished after delivery. That is the most expensive production model I have ever heard of.
And it has been running continuously for approximately 1 billion years, without missing a transcript, so...
Fine, fine. It works. I respect it.
After splicing, the mature mRNA is exported to the cytoplasm, where the ribosome reads it. This is translation.
Translation, reading RNA language and building protein language.
Exactly. And this is the point where people confuse transcription and translation. So let me be precise. Transcription, DNA to RNA, one nucleic acid language copied into another. Translation, RNA to protein, a completely different language. Nucleic acids converted into amino acids.
These being genuinely different processes that happen in different locations inside the cell, which is apparently not communicated clearly in schools.
Transcription happens in the nucleus. Translation happens in the cytoplasm, separated in space and time, different machines, different rules. The ribosome reads the mRNA 3 letters at a time. Each 3-letter combination is a codon. Each codon specifies an amino acid. Transfer RNAs, each carrying the correct amino acid, match their anticodon to the current codon. The ribosome forms a peptide bond and advances, one amino acid added per step until a stop codon.
How fast does this happen?
Approximately 15 to 20 amino acids per second, per ribosome. Your cells typically run many ribosomes on a single mRNA simultaneously, a structure called a polysome. A human cell contains millions of ribosomes running continuously.
15 to 20 per second, per ribosome, with millions running.
And they are accurate. One error per 10,000 amino acids, approximately, before quality control mechanisms. For context, a typical protein is 300 to 1,000 amino acids long. The ribosome almost never makes a mistake.
A machine I cannot see is making 20 decisions a second and getting them right. I take 10 minutes to decide what to have for lunch.
That is a fair observation.
The chain then folds. The folded 3-dimensional shape determines everything: what the protein binds, what reactions it catalyzes, what signals it transmits. Shape equals function.
And misfolding is a catastrophe. Alzheimer's, misfolded amyloid beta. Parkinson's, misfolded alpha-synuclein. Huntington's, a polyglutamine expansion that makes a protein fold into an aggregating clump. Some of the worst neurological diseases are fundamentally folding failures. The sequence is fine. The shape went wrong.
So the target is the fold, not the gene. Structure-based drug design. I've reviewed 12 companies in this space in the last year. 4 are worth calling back.
The field has existed for 40 years, but yes, the principle is the right one, which is also why I have been testing whether small molecules can rescue a misfolded target in my research unit. 2 cell lines are stable. The third we do not discuss in front of guests.
We literally have no guests, but okay. Okay, DNA to mRNA to protein. The central dogma. But here's the thing I cannot shake. Every cell in my body, skin cell, neuron, liver cell, has the same DNA, the same 3 billion letters. So why aren't they all the same cell?
Gene regulation, epigenetics.
This is where it actually gets interesting. This is my favorite part of biology, and I have never been able to say that to anyone who didn't immediately look slightly frightened.
Almost every cell carries the same genome. The difference is which genes are active. A neuron has activated genes for electrical signaling and synaptic function. A liver cell runs metabolic and detoxification programs. A muscle cell runs contraction machinery. The library is identical. Different chapters are open in different rooms.
So epigenetics is access permissions on a shared drive.
Cell-level identity and access management. I have a name, EpiGovern, per cell licensing. I'm writing this down.
You cannot put a SAS layer on histone acetylation.
What I am hearing is that nobody has yet.
2 core mechanisms. First, histone modification. The spool proteins can be chemically tagged. Acetylation generally opens chromatin and activates genes. Certain methylation patterns compact it and silence genes. Second, DNA methylation. Methyl groups attached directly to cytosine bases suppress transcription in that region. These marks don't change the sequence. They change the access.
And these marks are, what, permanent?
No, that's the point. Some persist for decades. Others shift in response to environment, nutrition, exercise, stress, sleep patterns, aging. Your epigenome is dynamic. The same genome reads differently depending on what's happening to the organism.
Wait, so my lifestyle is actively rewriting which genes my cells can read?
Yes.
Right now?
Yes.
That is genuinely, I need a moment. That is a lot.
It is mildly difficult to convey how different this is from the conventional "your DNA is fixed at birth" framing that most of us grew up with.
That framing is not wrong exactly. The sequence is fixed, but the sequence is only part of the system. Identical twins start with identical genomes. By middle age, their epigenomes have diverged significantly. Different diseases, different biology, driven by different environments and experiences.
Identical starting code, diverging output. That's a deployment environment problem. I've seen this. Different infrastructure, same binary, different failure modes.
Precisely. The genome is not a fixed program. It's a program plus a runtime environment. And you can edit the runtime. DCAS9 fused to a methylase domain, targeting a specific promoter. You can change which genes a cell reads without changing a single letter of sequence. I have 3 cell lines running exactly this protocol right now. 2 are behaving correctly.
And the third?
The third is technically autonomous. We're in a dialogue.
It is deeply alarming that that sentence was delivered with the same affect as everything else you say.
I am always this calm. It has nothing to do with threat level.
My grandmother changed what she ate after 60. She said the body was listening. She lived to 94.
Patar, is that an epigenetics testimonial, or is it completely unrelated?
Probably both. I think she was mostly just talking about soup.
Can we sequence her?
She's 94.
Biobank. Longitudinal data. Someone in this room should be calling her.
We have been from 4 letters, through the double helix, through 2 meters of DNA per cell, through ancient viral DNA we've turned into placenta, through ribosomes making 20 decisions a second, to the genome running like adaptive software in every cell in your body. Patar. You have said approximately 8 things this entire episode. What is DNA? Patar.
DNA is a message written in 4 letters. The cell keeps the original locked up and makes copies of only the sections it needs. Ribosomes read those copies and build proteins that do everything: structure, enzymes, signals, all of it. Epigenetics is the permission system. Same message, different access in different cells. The whole thing is just information. Stored, copied, read, and controlled. And 8% of it used to be a virus. That's it.
Patar, 6 sentences, the whole episode in 6 sentences, including the virus.
She has accurately described the central dogma: chromatin accessibility, differential gene expression, and endogenous retroviral integration. I actually find that quite remarkable.
Same message, different access. That is the best summary of epigenetics I have ever heard. I am writing it on the wall of my research unit.
It's a shed, Kelpie.
The wall is real.
Same message, different access is a phenomenal pitch framework. Identity architecture. I'm using this. Full credit to Patar. Series A closes Friday.
That is the Krusty lab. Every single cell in your body is running this right now, reading sections of a 3 billion letter message, firing ribosomes at 20 amino acids a second, and managing which genes it can even see. The system is 3.5 billion years old. Part of it used to be a virus. It mostly works. Mostly.
I would personally rate it very highly. But I may have lost objectivity. I've been inside it for several weeks now, conceptually.
It is mildly comforting that nobody here is qualified to offer a stronger guarantee than mostly.
The dog is fine, for what it's worth.
The neighbor's dog named DNA?
He mostly works, mostly.